Disinfection is a crucial stage in water treatment processes before water can be distributed via the drinking water network. Now, what characterizes this final stage in making water drinkable, and why is it essential? Well, first, we will precisely define disinfection. Then the different methods used for disinfection will be studied. Water contains a high quantity of microorganisms. Now, amongst these are algae, bacteria, fungi, viruses, protozoa, and spores. Now, some of these microorganisms are harmful to health. Now, these are called pathogens. Disinfecting water consists of removing or deactivating these microorganisms in order to avoid their development or reproduction. And disinfection may be carried out in several ways. One, physically, generally by using ultraviolet light, or two, chemically, by creating chemical reactions using compounds such as ozone or chlorine. Ultraviolet or UV light destroys bacteria, viruses, fungi, algae, and yeasts by altering their DNA without using a chemical agent. When disinfection with ultraviolet light is used, the water that is to be treated must not contain any suspended solids or compounds likely to absorb UV light, such as iron, for example. Turbidity must be less than 0.3 nephelometric turbidity units or 0.3 NTU. UV light is characterized by a certain range of wavelengths. Now, this wavelength is written using the lambda symbol and commonly expressed in nanometers. Now, three main types of UV are differentiated: UVA, UVB, and UVC. Now, only UVC light, the wavelength of which is between 200 and 280 nanometers, is capable of destroying pathogenic microorganisms. Now, their removal is optimal at a wavelength of 253.7 nanometers. Now, UV light is produced by mercury and argon vapor lamps. Now, each lamp is placed in a quartz duct transparent to UV light, which protects them from coming into contact with water. Now, an electrical shock is applied between two electrodes in the lamp, thereby exciting mercury atoms which emit ultraviolet light. Water flows in fine layers in the middle of the quartz ducts. Now, these ducts are laid down in parallel or perpendicularly to the flowing water, either vertically or horizontally, in reactors. And these reactors are in uncovered channels or enclosed and pressurized. Now, in the case of drinking water, closed pressurized reactors are used. Chemical disinfection may be carried out in several ways. Now, currently, the most commonly used reagents are chlorinated compounds and ozone. After dissolving, the chlorinated compound will form hypochlorous acid, which is written as HClO, which is a disinfectant that prevents bacteria and protozoa from growing. Now, chlorine has a considerable advantage. It will stay in water long after it has been injected into it, so it is said to be residual. Moreover, it is easy to diffuse continuously, which enables its concentration in water to be checked and monitored. However, it does have some disadvantages, such as the formation of byproducts that are toxic for human beings, such as chlorites or chlorates, which have potentially harmful effects on the liver or the thyroid gland. Lastly, using chlorine also requires ensuring that pH is not too high, as otherwise its effectiveness is diminished. Ozone is another major compound that is used to disinfect water. Now, it acts directly and indirectly. Direct disinfection corresponds to the removal of the toxicity caused by pathogenic microorganisms in water. In this case, ozone damages the cell wall of the bacteria present in the water. The cell breaks under the effects of internal pressure within it, also called osmotic pressure. The bacterium is thereby killed. Indirect disinfection or oxidation corresponds to the removal of toxicity caused by dissolved micropollutants in the water. Now, in the case of water with a pH of neutral, near to 7, and in the presence of organic matter, ozone can form hydroxyl radicals, written as OH, that plays an important role in deactivating the micropollutants. What you need to remember about disinfection. Well, disinfection enables pathogenic microorganisms, pathogens, to be removed or deactivated. Also, physically, disinfection is generally performed using ultraviolet light. And lastly, chemically, a chlorinated compound or ozone is used.